class lecture for the week of September 24th and September 26th for the fall semester History of Life. This is Dr. Sheffield. So please make sure you watch this video and ask questions over email or in person when I'm back if you don't understand anything. So we left off talking about the origination of unicellular life, that is single-celled organisms called prokaryotes. So today we're going to be talking about the evolution of the next stage, multicellular life. So prokaryotes, the group that we've already talked about, are divided into two large groups, archaea and bacteria. Now, we don't really need to know the differences between them for this class. It mostly deals with the chemistry of the chemistry of their bodies. But archaea has groups like halophiles in it. Phile means love. Halo means salt. So archaea has a lot of organisms that really love extremely salty regions that most organisms can't survive in. All right, so there was archaea, or an image of where archaea would live, and here's some bacteria. So here is the large tree of life. Now, you don't need to know all of the words here, but I want you to pay attention. All right, so what we have is a common ancestor. And along that common ancestor down here, we see this split between bacteria and archaea and eukaryota. Okay, so bacteria and archaea are the prokaryotic organisms we're going to be talking about. Eukaryota is all the way up here, sister to archaea. You see that group here. And it includes things like slime molds, animals, fungi, plants, etc. So, where do you see yourself here? You are right here under animals. All right? So you're actually pretty closely related to a lot of these groups. Now, that might seem strange, but this tree is giving you a very broad picture of life on Earth, okay? So you're not seeing all the different relationship breakdowns like you would on a very narrow scope. So to review from last class, fossil prokaryotes are pretty difficult to identify because they frankly kind of all look the same in the fossil record. We've got some that are like blob-shaped, some noodle-shaped ones. We really can't get further than that without having that genetic data. But we do know that they exist. And how we know what they're doing at this time is by chemical signatures. So remember we talked about we see photosynthesis showing up and methane production showing up a little bit more. Okay, here's those examples of the bacteria. Now, we're going to talk about the next stage of life here. Stromatolites. So what you're looking at right here on this picture on the right is a stromatolite. These are layered biofilms. So how this works is we have a mat of bacteria that might lay down on the ocean floor. It'll get covered with a little bit of sediment, all right? So because there's a bunch of sediment like sand in the ocean. And then another mat will lay down, and that process will repeat until you have this giant layered mat of disgusting bacteria, all right? Now, while these are mostly unicellular, again meaning one cell, some prokaryotes can form into giant piles of these. And these are going to be called biofilms or stromatolites. We're going to call them stromatolites for now. So the oldest stromatolites have been found in Australia, and they're dated to about 3.5 billion years ago. So this is what they look like. You can see all the little layers right in there. And they're interpreted as a type of green algae, cyanobacteria. And we already saw a picture of that cyanobacteria last class. It was those little circular blobs. All right, but that's what it looks like in its full form. So these are pretty common Archean fossils, and there's some alive today. So one of the best places in the world to see them, and one of the most protected sites in the world, is Shark Bay, Australia. So here are our stromatolites still growing, still alive today, and we believe that they are fairly similar to the ones in the Archean. There's probably some differences, but for the most part, they're similar. But Shark Bay, Australia is incredibly famous. It's a World UNESCO site, meaning you absolutely cannot damage the property in any way, shape, or form. And it's so famous, they actually have a mascot stromatolite walking around to tell you all about the site. This is not a lie. So, eukaryotes. Eukaryotes, by their definition, are organisms with a cell nucleus containing their genetic material, meaning they have membrane-bound organelles. All right, so you are a eukaryote. All multicellular animals are, all multicellular organisms are eukaryotes. They can be unicellular, though it's not terribly common, but all multicellular organisms are eukaryotic. Okay? So again, here is our tree. all of the organisms on this branch here, this brown side, are either unicellular or multicellular, but this is the only place multicellular organisms will show up. So the earliest eukaryotes in the fossil record show up in the Proterozoic. So we already went from the Hadean eon, which was named for Hades, the Archean, which was named for the first evidence of life, and now the Proterozoic. The Proterozoic is going to be the first evidence of slightly more complex life. So these are just words on our time scale. Again, you don't need to memorize these words, but you should at least know that they mean time. If I say it happened in the Proterozoic, you should know that that is a time unit, not a country you've never heard of before, okay? So this is what they look like, eukaryotic algae. Now you're looking at this saying, okay, sure, Dr. Sheffield, whatever you say, it's eukaryotic algae. How do we know? Well, that is an excellent question. So how we know it's eukaryotic and not prokaryotic is by organic compounds called steranes. You don't need to know the word steranes. You can just call it organic compound or chemicals that were found in these rocks. These steranes are only produced by eukaryotic organisms today. And by the understanding of uniformitarianism, if eukaryotes are the only organisms that produce steranes now, we assume that if we find these in the past, they were likely eukaryotic as well. So now this begs the question, why would we have two different types of cells? Why would we have prokaryotic and eukaryotic cells? Why would we switch when most of the organisms alive today are prokaryotic, right? There's way more bacteria than there are humans, to be clear. So that's a really good question. Eukaryotes have specialized cells, organelles, They're subunits of cells. Some examples are things like the nucleus, the ribosomes, the chloroplast, the Golgi bodies, and the very famous mitochondria, the powerhouse of the cell. Now, some of these subunits seem to represent remnants, meaning leftover pieces, of early symbiotic relationships. Okay, so I want you to remember the word symbiotic relationships. We'll go over it here. A symbiotic relationship. Now, we've already learned from synapomorphy and sympatric speciation that SY means together. Okay, so biotic means living. So we've got organisms living together, okay, interacting together. That's what that word means. So a symbiotic relationship is where two organisms have to interact with each other in real life, okay? So for example, here's a clownfish and an anemone. Ooh, try saying that 10 times fast in a microphone, right? So the clownfish is actually cleaning off bits of the anemone, but you know what? It's not really doing anything super important to the anemone, but the anemone is providing protection, right? It gives the clownfish a place to live, a clownfish a place to hide from predators. So the clownfish does do a little bit, right? But not really. There's some other really great examples like, there are certain types of birds that clean out crocodiles' teeth, and in exchange, those crocodiles don't eat those birds for breakfast. All right, so there's different types of symbiotic relationships. Some have positive effects, some have negative, some are neutral. It doesn't really matter. Humans and Netflix are not technically symbiotic relationship, though I suppose you could argue it. I am joking. So this is going to bring us to a very famous scientist, Lynn Margulis. So she died in 2011, and she was an American biologist, and she hypothesized about something called endosymbiosis, which has now been elevated to a theory, meaning it answers the question of why we have eukaryotic cells in the first place. And her original hypothesis, which has been supported by multiple tests on the order of thousands to tens of thousands of scientific inquiries, suggested that perhaps some of our organelles were actually their own prokaryotic cells in the first place. So mitochondria and chloroplasts are the two big examples. You don't need to know their proteobacteria, their cyanobacteria name, but you do need to remember at least the word mitochondria for me, and certainly endosymbiosis. So let's break this down. What does this have to do with a symbiotic relationship? So cyanobacteria, along with other types of bacteria, photosynthesize, meaning they make their own food. They they use water and carbon dioxide and sunlight to make oxygen and some sugars. The sugars are their food. So the idea is other prokaryotes essentially swallow kind of engulf. Swallow is not the correct word here. Swallowing is a very active action that you take. But imagine with me here, use a little bit of hyperbole here, that they swallow these delicious cyanobacteria. So essentially they just engulf it into their body. Now this is a process in biology called phagocytosis, cells eating other cells. All right. So you don't need to know that word, but it's a fun word. So I thought maybe you'd be interested. So they wrap their membrane around them and engulf them. And when they do that, the prokaryotic cell that was just engulfed isn't really harmed. It's just now within another cell. So what does this have to do with symbiosis? Well, the cyanobacteria that was just engulfed is now protected by a larger cell. It has its own membrane and now a second membrane to protect it even more. Cyanobacteria can duplicate within the hosts of other cells, so it can just keep duplicating. So things like a mitochondria, if this happened, the mitochondria could pretty much pretend like it wasn't engulfed in a cell and it could just keep living on its own. All right? So chloroplasts have their own double membrane, just like mitochondria do today. So a chloroplast is something found only in plant cells. Mitochondria is found in all of our eukaryotic cells. All right, but these organisms have a double membrane all of their own. So how is this symbiosis? What is the other organism doing? So remember, let's go back to the example of we have one cell swallowing the other. The one that was swallowed now has some protection, and it has an extra membrane to keep protecting it. Now the cyanobacteria has a cushy place to live, we've just said that, but the cyanobacteria is photosynthesizing. So the cell that engulfed it is getting some of the leftover sugars that the photosynthesis is producing. So it's actually a mutual beneficial relationship here. Both organisms are being helped. And if that's the case, if both organisms are being helped, why would that relationship stop? So mitochondria probably had a really similar beginning to this as well. So chloroplasts definitely did this. Mitochondria likely did it as well. Now, one of the reasons that we know this is both mitochondria and chloroplasts. Again, we just talked about how they have their own membranes along with the cell membrane, which other organelles don't have, but they also have their own genetic material that's separate from the surrounding cell. So mitochondria has its own special type of DNA. We call it mitochondrial DNA, mDNA for short. Chloroplast has its own too. And further, both organelles replicate like bacteria. And what that means is the mitochondria and the chloroplast can actually reproduce itself in the cell without any input from the nucleus or any other parts. So the cell itself has to have a whole bunch of different systems in play to replicate itself. Mitochondria can just do it on its own. So this theory has quite a lot of support. Now this image here shows you exactly what's happening. So we've got our photosynthetic and oxygen-breathing bacterium here, and they essentially get tucked into the cell, and over hundreds of thousands of generations, they lose that ability to live on their own. And that might seem a little strange, but this actually happens a lot in nature, and we can see that today. So coral, for example, coral reef is beautifully colored if it's thriving and healthy, unlike most of our coral reef today. But because it's so, let me rephrase, the reason that it's so colorful is actually because it's covered in a small microscopic organism, photosynthetic organism, called zooxanthellae. And the zooxanthellae photosynthesize, and they give the coral the beautiful color. All right? So there's a beneficial relationship there for a number of reasons. But the long part, or the short part of the story, the moral of the story here, is when those zooxanthellae can't survive, the coral actually dies with it. The coral has lost the ability through evolutionary processes to survive on its own. Okay? So when we talk about the idea of going from prokaryotic cells to eukaryotic cells, the idea is that one prokaryotic cell essentially swallowed another cell, and that relationship persisted into making eukaryotic cells. This is called the theory of endosymbiosis. Endo means inside, symbiosis means a relationship between two organisms. And you should definitely know this. Okay, so we're going to switch a little bit here. So to review, in the Archean, we had very little free oxygen. What that means is we had oceans, right? And oceans obviously have oxygen in it because oceans are made of water, which is H2O. But free oxygen is oxygen that's not bonded with hydrogen, okay? And Earth's atmosphere contained very little of it. The original bits of it definitely came from volcanoes, and we're going to fill in the rest here a little bit later. So atmospheric oxygen is going to come from 3 different sources, okay? The first of which is rock weathering. So when we weather rocks, we can release oxygen. For example, here is a sandstone shown here. The sandstone is made of quartz, SiO2, silicon dioxide. All right, quartz is the same thing as the pebbles you see on the street, amethyst, rose quartz, all that stuff is the same thing. So when we weather it, it can actually release the oxygen that's bonded to the silica, and we can get minor amounts of oxygen released into the atmosphere this way. A secondary way is through photolysis. Photo means light, lysis means breaking, so it literally means the breaking of light. And how this works is it's a chemical reaction in which a molecule is broken apart by light photons coming from the sun. So we can do this by the breakdown of water vapor or other gases like carbon dioxide, etc. Okay? And this is actually how we're going to get the formation of ozone. And if you'll remember, the only reason we have an ozone layer is thanks to the magnetic field, which was formed during the iron catastrophe. And finally, photosynthesis. This is the one we're likely most familiar with. Photoautotrophs, which is a $1,000 word right there, but it just means that organisms are making their own food by using light. So they take in carbon dioxide and water, plus energy, sunlight, to make sugar and oxygen. So this is where we're really going to see the most of our oxygen coming from. Okay, now I've shown you a leaf because that's what we're most familiar with, right? Plants doing this. But at the time, there were no plants. There's just bacteria. So we start photosynthesizing about 3.5-ish billion years ago. We start introducing oxygen into the atmosphere. So it's not a switch that's flipped. We don't go from no oxygen to enough for humans to walk around. But how do we actually estimate how much oxygen was around without being there? Well, there's some really excellent ways that we can do this, so I'm glad you asked. And this is going to take us to an event called the Great Oxygenation Event. Dun, dun, dun. I'm going to take a quick water break. Give me a second. All right, so the Great Oxygenation Event. Here you see a beautiful rock, and it's got some really gorgeous layers to it, all right? Hopefully you can see these gorgeous red layers. These are caused by an element called, you guessed it, iron. Very nice. So we're going to talk about this. This is a geologic timescale. Today we are here. So September 20th, the day that I'm recording this, we are in the middle of the Holocene, which was nothing geologically speaking. It was, it's recent. Today, we're spending most of our time in the Archean all the way up to the Proterozoic. And again, you don't need to memorize these words. You just should have at least understanding that these words mean time. Okay, so we're in the Archean, the Proterozoic, the Hadean is cut off. Sorry, it's down here. So this is the Archean world. It's very soothing to look at, right? So this is painted by a science artist, Peter Sawyer. It hangs in the Smithsonian Museum of Natural History. And you're looking at it and you see these beautiful volcanoes in the background. You see a very calm, rippling lake. You see some stromatolites here. But how much oxygen is there? Excellent question. So scientists hypothesize that the atmosphere is almost entirely lacking in free oxygen at the beginning of the Archean. How do I know? All right. So first, this is just a picture of pebbles, but it actually tells us a pretty important story. And this story has to deal with an element called pyrite, which you do need to know about. Okay, so pyrite, also known as fool's gold. You might recognize it by fool's gold. So here is pyrite everywhere. Fool's gold tells us a really important story. You don't need to memorize its chemical formula, but there's a pretty good hint in it if you do. Its chemical formula is FeS2. All right, so it's made of iron and sulfur, FeS2. Now, if you've ever left your bike in the rain and your parents have yelled at you, you'll know a little bit about pyrite. So your parents yelled at you because you left your bike in the rain, which caused the iron in your bike to oxidize, otherwise known in normal human terms as rust. So this is pretty important because pyrite, being made of iron, usually rusts in the presence of free oxygen. It won't form. It can't form. So why this is important is pyrite usually forms shapes like this, a beautiful cube. This is called isometric if you're interested or a geology major. So this is what freshly formed pyrite in the presence of no oxygen looks like if you look at it real close. It's cubic. So let's take a look at this again. Hopefully you can agree with me that this is not cubic. It is in fact quite round. Now how this would happen, if you imagine with me, usually I do this demonstration in class, so that's not going to happen. Let's take a hunk of clay, okay, a hunk of clay, and let's make that clay into a cube. Let's start tossing it around the classroom. So I'm going to toss it to the front row, who's going to toss it to the middle, to the back, all over the place. Every single student's going to toss it at some point. Over time, that cube is going to get duller and duller edges. It's going to round down essentially into something that looks like this. So the interpretation of rocks that look like this, and this rock is almost 3 billion years old, it's about 2.5 billion, okay, is that the pyrite was actually being knocked around on Earth's surface. This is what rocks look like if they've been tossed around in a river, for example, for a long period of time. They get rounded down. So if this pyrite has been knocked around on Earth's surface for thousands to millions of years and hasn't rusted away from billions of years ago, we can estimate that it was likely there was very little oxygen in the environment. All right, so just to summarize that again, the pyrite has very rounded grains, which means it's been knocked around on Earth's surface for a really long time. The fact that it didn't weather away is an indication that there was no oxygen, because pyrite is made of iron, and that's what iron usually does in the presence of oxygen. It rusts away. So here's another clue. This is a rock. Again, this is a banded iron formation. Okay, so you can see why it gets the name banded. It's got these beautiful bands of iron. So let's say some of these iron atoms made it. So they were traveling around on Earth. They were going through rivers. They never actually encountered any oxygen. So all rivers eventually dump into the ocean. Okay, that's how rivers work. They erode and erode and erode. They wear themselves down until they get to an ocean basin, like the Mississippi River. And they dump a whole bunch of sediment in it. So that sediment is going to contain a lot of iron if there's no oxygen in the atmosphere. And so we just have lots of iron built up in the rivers going into the oceans. That iron will eventually be deposited in the ocean. Now, use your remembering skills. Who is living in the ocean at this time? Hopefully you're saying bacteria. Okay, and what is that bacteria doing with their life? Well, they're not going to college. They're just photosynthesizing, right? So if they're photosynthesizing, they're making what gas? They're making oxygen. So what's happening to the iron in this picture? It's interacting with the little bits of oxygen that are in the ocean, and it's rusting that iron. So just because there is no oxygen doesn't mean there's no life. Oxygen is not a requirement for life, and in fact, the vast majority of life forms on Earth don't need oxygen, okay? So there are prokaryotes and some eukaryotes. Many eukaryotes don't need oxygen either. So stromatolites are thriving. Bacteria is thriving during this time, even with very little oxygen. But that's going to bring us to the Great Oxygenation Event. Now we call it that because it was a really good thing for humans, right? But here we have the timeline. So we have a red and green line. My apologies for those of you who may not see red. But this red line here, this is the top line. The green line is the bottom line. So all you need to see is that around this time, oxygen did rise. Did it rise a lot? No, it didn't. It didn't rise very much at all. But it went from 0 to quite a bit more than 0. And as you're going to see here, it's going to continue to rise for a really long time. So this is today, September 20th or so, 2019. You can see that oxygen's even been higher in the past. We'll talk more about that later. But if what I'm telling you is true, that oxygen rose 2.5 billion years ago, what would we see? Well, first, we would stop seeing rounded pyrite, but we would start seeing oxidized beds. So this is an example of an oxidized bed, and what I mean by that is there was iron in the sediment that reacted with oxygen, so it rusted. So just like your shovel or bike that was left out in the rain, this is what you would see. Now, these pictures aren't from the Archean. These are very common rocks you might see out west in like Nevada or Colorado. These particular ones are Triassic in age. They're pretty much all Triassic. So next time you're out there like on a family vacation, you can impress absolutely nobody by yelling, "Hey, look, Triassic rocks." But there you go. So we're also going to see the presence of a new type of mineral, and that mineral is called gypsum. You don't need to memorize the chemical formula of gypsum, although I assume you'll all want to. CaSO4H2O. Depends on how many molecules there are. Usually it's 2H2O, okay? But gypsum has to form with a heck of a lot of oxygen. As you can see, it needs 4 oxygen atoms, all right, to bond with the sulfur. So we have CaSO4H2O. Gypsum cannot form without a lot of oxygen. So around 2.5 billion years ago, we see a switch to a lot of gypsum being formed, okay? So gypsum is kind of like halite, which halite is table salt, essentially. It forms in evaporating environments, so a lot of the times near seas that are really warm and humid, things like that. We also see a disappearance of pyrite in a lot of areas, and we see pyrite only forming in low oxygen areas. So where are we going to see low oxygen areas? Well, we're going to see them in a place called anoxic. A means without, oxic means oxygen, so no oxygen, parts of the ocean. Those types of places still exist today, and we've had them throughout all of Earth's history, areas where there's just like no oxygen in the ocean, so no free oxygen circulating around. So this is an ammonite. It's something that lived about the time T-Rex did, so it's not an Archean fossil, but it's covered in pyrite, meaning it sunk after death to one of these areas with low oxygen. So now the second question. Can banded iron formations form when there's a lot of oxygen? They can't, all right? So what happens again is banded iron formations, we have tiny bits of iron coming from rivers being dumped into the ocean. If there's a lot of oxygen, they're going to rust before they ever even manage to get anywhere close to the ocean. So after banded iron formations, which we'll call BIFs, not to be confused with the villain from Back to the Future, do after the GOE, which is the Great Oxygenation Event, sorry for all the abbreviations, well, they disappear. They don't show up. So again, this doesn't mean we went from just bacteria to T-Rex levels of oxygen here. This isn't how it worked. Oxygen was a very slow increase. Okay, so it didn't really increase that much. Now, I can't give you a numerical amount of how much it increased. We have a pretty wide spectrum of measurements here. They're proxy measurements, meaning we can measure the amounts of sulfur and iron in rocks to estimate how much oxygen there is. All right, it's like proxy data is like if I wanted to know the temperature outside, I could look in the classroom to see if students were wearing coats or tank tops. It can give me an indication of what we're looking at, okay? So these ranges are pretty broad, but they suggest that oxygen was at about 10 to the negative 57th to 10 to the negative 5th present atmospheric levels, meaning how much oxygen there is today. So regardless of how much there was, we know it was a lot less than what today was. So why did it increase? So photosynthetic organisms, again, cyanobacteria, take CO2 out of the atmosphere and they produce oxygen with it. This is the photosynthetic equation. You don't need to memorize it, but you should be able to use this equation to tell me what's happening. Okay? So is this the only reason why oxygen increased? It's actually not. Okay? Now here's going to be a quick chemistry review. You're not going to be tested on it, so don't worry too much. But just remember that things like C6H12O6, we can reduce it. So we can reduce each of these numbers by 6 because they're all divisible by 6. So we can make that CH2O and it would mean the same thing, okay? Don't forget that. So oxygenetic photosynthesis is only part of the equation here. So something else had to happen for all of this oxygen to increase. And that is for oxygen to increase in a large amount, like we will need to be able to get anywhere near humans needing to breathe, we need to remove organic carbon from the oceans. So when organisms die, their body just sits in the ocean, and that's organic carbon. Just like in the middle of a swamp where all those trees, they're just releasing and decaying their leaves, that's releasing organic carbon. So how are we going to do that? Well, this is a geology class, so you can imagine that the answer is going to have to deal with geology somehow. And you're right, it's going to deal with plate tectonics. So this is a delta system. This is a delta in Alaska. All of these small river tributaries are bringing tons and tons of sediment, you can call it dirt, into the oceans. And if we have enough of it, we can actually bury some of the organic carbon. So how we're going to do this, if we go back a couple billion years, we have a lot of supercontinental formation. Now, everybody remembers Pangaea, the supercontinent, but many people don't recognize that there were many supercontinents that formed before Pangaea. There was Rodinia, there was Ur, there was Kennerland, the one we're looking at here now on the screen. They don't really mean much to us right now, and that's okay. But imagine that the Earth is just a giant bowl of milk, and that these continents that we're looking at are like giant pieces of cookie. So if you slam these cookies together over and over again, those cookies are going to break down. Okay, there's going to be a lot of crumbs. Those crumbs are going to go into the ocean somehow. All right, and again, the ocean is milk. So when we have lots of plate tectonic activity, lots of things smacking into each other, we're going to have more and more and more rocks or cookies breaking down over time. So here's a modern example. This is the Ganges-Brahmaputra Delta. All right, it is attached to the Himalayan mountains, one of the largest mountain regions in the world, and the largest mountains that are above the surface of the ocean. Okay, their annual sediment load, the amount of sediment, dirt that it can move, is 1 × 10⁹ tons per year. So it's moving a whole bunch of sediment. So now if you imagine, if continents are all slamming into each other, we're going to have a lot of sediment being put into the oceans. So plates are going to pull apart. They're going to slam together. They're going to make mountains. Those mountains are going to break down over time. We're going to have lots of sediment entering the system. And when I say sediment, what's in these sediments are things like phosphorus, nitrogen, oxygen, and potassium. Essentially, nutrients that are going to help life flourish. Do you see what's going to happen here? Now we also have a lot of sediment going into the oceans that can bury that organic carbon that's being left behind by dying bacteria. So this is the equation here. We are reducing this carbon dioxide by photosynthetic organisms like bacteria using it. All right? This carbon here on this side, remember this was the C6H12O6, that's sugar. This carbon is being removed from the surface oceans. This is a byproduct of the plant. And when the photosynthetic organism, when that photosynthetic organism dies, it's released back into the system. So to get rid of it, we're going to bury it with plate tectonics. We're going to take something like the Ganges-Brahmaputra Delta and quite literally bury it. And finally, all of this oxygen is going to start to be released. And we're releasing it again through photosynthetic activity. So if our model of reduce, remove, release is happening during the Great Oxygenation Event, we should see this preserved in the rock record. As you can imagine, excuse me, I forgot where I was going with this. So this is going to be called the 3 R's. Reduce, remove, release. When I ask you about the 3 R's, please do not tell me to reduce, reuse, recycle. I do appreciate that, and I do encourage you to do that in your own lives, but it's not the 3 R's I'm looking for. So please do not just Google what are the 3 R's. You will not get the correct answer. So this is going to bring us to a system called carbon isotopes. So an isotope is a system that forms with the same number of protons but different neutrons. So carbon has 6 protons and 6 neutrons in a regular day. That gives us carbon-12. 6 + 6 is 12. This is going to rely on some basic math skills. However, we can have an isotope, and that means that it'll have a different number of neutrons. Never protons, only neutrons. So carbon-13 has 6 protons and how many neutrons? 7. 6 + 7 is 13. All right, which one is heavier? Hopefully you're saying 13, and it's heavier by 1 neutron. So what type of carbon will photosynthetic organisms preferentially, meaning they will want to take it in? 12 or 13? They're going to want to take in 12. And the reason for that is 12 is a little bit easier to break down. So if you imagine with me that we're all in the Olympics and we're all running a 50-meter dash. Before the event, we're given the option of going to Golden Corral for an all-you-can-eat buffet breakfast or a hard-boiled egg and a Greek yogurt. Which one are you going to pick? You're probably going to pick the hard-boiled egg and yogurt, right? Because you don't want to vomit all over national TV. If you eat so much food for breakfast, it's going to be really hard to break that down. And then if you go running, oh dear Lord, you're going to, throw up everywhere. So imagine this with photosynthetic organisms. C12, the lighter of the two, is going to be preferentially removed by photosynthetic creatures, which means they'll still take in the C13, but they're not going to break it down. So it's going to be released in their body when they die as those sugars. So in times of high carbon burial, that's what the phrase is called, we see more C13 in the rock record. In times of low carbon burial, or in times of low photosynthetic organisms, there's more C12 than usual. Okay? So isotopes don't occur in 50-50 percentages. I can't say, okay, there's 51% C13, we must have more photosynthesis. They're very minor shifts, and they're not shifts I expect you to understand numerically. You're just going to look at a graph and see if it's going up or down. If you take an isotope geochemistry class, of course that will change. So let's add on to this. What is the temperature like with very few plants making oxygen? It's going to be pretty warm outside, right? Because that carbon dioxide is not being pulled out of the air. That's why today we're trying to plant as many trees as possible to remove some of that carbon dioxide that we've been putting out. So the more plants there are, the more photosynthetic organisms, there'll be more oxygen being produced, so it will be cooler outside. Okay? So very important. C12 and C13 are not the same thing as carbon dioxide. Okay? C12 and C13 are just isotopes of carbon. Carbon dioxide is a gas. Carbon dioxide is a greenhouse gas. The more CO2 there is, the warmer the temperature. CO2, however, can be made with either C12 and C13 because that's what the photosynthetic organisms are pulling out. They're pulling CO2 into their bodies, regardless if it's C12 or C13. They just only use that C12. So the more C13 we see in the rock record, that C12 is used, what's left behind is the C13. The more C13 generally indicates more photosynthesis. So high carbon 13 actually indicates lower temperatures. Okay? High carbon 13 indicates lower temperatures. So here, this is our carbon 13 graph. Okay, so the change, that's delta, the change of carbon 13, and there's the unit. All right, parts per mole. So here we have a positive excursion, that's what we would call it. You can just call it an increase. A huge increase of carbon 13. What event do you think we're looking at right here? If you guessed the great oxygenation event, you would be absolutely correct. We have our carbon 13 spiking super high. Now you can see that it crashes pretty quickly. All right, and there's a reason for that. So once we first introduce oxygen as a really big, or as a wide amount of gas into the atmosphere, Earth kind of goes a little crazy for a little bit. And as you can see, it's going to take some time to stabilize out. You can see all of these events right here. Okay? So we're not going to worry too much about that, but you'll see that carbon 13 doesn't stay stable in the rock record. And that definitely means that photosynthesis doesn't stay stable in the rock record as well. So let's summarize. We have our red and green lines here. This is the red, the top line, the green is the bottom. How much did oxygen increase? We're still not entirely sure. We know it didn't increase a whole bunch, but we do know that it did increase. Scientists are currently working to better understand how much it actually increased using numbers. All right, so if you're interested in that, we can always talk about it further. So we can't measure oxygen isotopes directly in the rock record past about 400 million years. And again, this is quite a few billion years ago. And the reason we can't do that is after about 400 million years, they usually get a little wonky, so we can't actually use them. We have to use other systems that haven't been altered in any way. That's why we use sulfur and iron. Before the Great Oxygenation Event, we know there was very little oxygen before, because we have rounded pyrite. Remember, rounded pyrite means it's been tossed around on Earth's surface for a while. We also have the presence of banded iron formations. Okay, so this is where iron made it all the way to the ocean before rusting. We know there was more oxygen after the GOE because of the presence of this mineral here, which is called gypsum. Gypsum, the mineral formula being CaSO4, SO4 2H2O, I believe, okay, has a lot of oxygen in its system bonding with that sulfur. So we need lots of oxygen for gypsum to form. We also here have red beds. That's their nickname, red beds, because they're red. And they have interacted with the oxygen in the atmosphere. There's iron in there in this sediment here, and it's interacted with the oxygen. It has oxidized or rusted, turning it red. This is the graph that helps us understand this process. So this is photosynthesis. Again, this is C6H12O6. It's just been reduced by 6, so CH2O. So carbon dioxide plus water and sunlight will give us a product of sugar and oxygen. So organisms that photosynthesize will take in the CO2. That's how we're reducing it. And on the flip side, they're going to release oxygen. That's basic respiration in plants and bacteria. Now, this leftover product is going to get removed when we bury it in the rock record. So quite literally, we have rivers coming in with tons of sediment, and they're just dumping it into the oceans. The more dumping we have, the more of this is going to get buried. All right? Now, what's being broken down here is C12. What's being removed from this system here is C13. Organisms don't usually break down that C13. So they keep it in their bodies. All right? It's harder to break down. So the more C13 there is, the more photosynthesis there was at the time. And if we know how much photosynthesis there was, we can estimate how much oxygen there might have been. But we need both photosynthesis and the removing of carbon to actually allow oxygen to rise. So this is also technically called the oxygen catastrophe because there's lots of organisms where oxygen is toxic. So there was a major extinction event among anaerobic, meaning non-oxygen organisms around 2.5 billion years ago too. But because we're humans and we're biased, we really love oxygen, we call it the Great Oxygenation Event instead of the oxygen catastrophe. So this is going to bring us to the boundary of the Archean and the Proterozoic. So we started with the Hadean, then we moved to the Archean, which is where we've been for the past 2 days. Now we're in the Proterozoic. The ratio of carbon isotopes, specifically carbon-13, shifted towards more carbon-13. Meaning what? If we have more carbon-13, that means we have more organisms using that C12 up, that C13 is getting buried. So we actually have low global temperatures. All right? More photosynthesis means more oxygen's being released, which means lower temperatures. So, yep, global temps are falling, oxygen levels are rising. Nice work. So part of this increase, again, is because of photosynthetic organisms, but it's also because of plate tectonic activity. And both types of cells, prokaryotes and eukaryotes, are experiencing evolutionary radiations. This is a mega complicated phylogenetic tree. You do not need to worry about reading it. So by 2.5 billion years ago, Earth is still continuing to cool. This is a picture of one of the earliest supercontinents called Ur. Ur is a German root for original. That's where it gets its name from. So we think it was the first supercontinent, though we're not totally sure. So once it starts to cool down on Earth, plate tectonics are going to slow down just a tiny bit. So we're going to build up more continental crust, and plates are going to be moving a little bit slower. All right? We've got widespread tectonic events, meaning lots of mountain building, lots of supercontinents forming. Here's just another example of another one. This is the Kapval Craton that touches South Africa, Zimbabwe, parts of Namibia, though this graph doesn't really show that. All right? Just a picture of what these looked like in the past. So what do plate tectonics have to do with life? We already know a lot. Plate tectonics lower the temperature of Earth. Right? They get rid of some of that steam in the center of Earth. Steam isn't the right word, They get rid of some of that heat in the center of Earth. They release that heat onto the surface where it eventually cools down. Volcanoes give us water. They give us atmospheric gases. And plate tectonics give us mountains that weather organism or weather nutrients. So we get nutrients into the oceans. So plate tectonics are absolutely critical towards life on Earth. All right? So early supercontinents. Here is one called Rodinia. This is the most recent one after or before Pangea. This was about 1 billion years ago to about 750 million years ago. All of these are proposed combinations of what Rodinia might have looked like. Now you might be thinking, okay, these look really different. What are scientists doing? Well, part of this issue is it's pretty difficult to figure this out. One, a lot of these rocks have been metamorphosed, meaning changed through time, so we can't use those rocks. They've been destroyed through subduction processes, and some of them are just covered by trees, so it can be really difficult to find this data set. So there's a lot of different reconstruction ideas possible. This is done with paleomagnetism, by the way. So we look at the rocks to see if they are pointing magnetic north or south, and then make a giant jigsaw puzzle to recreate what these might have looked like. So this is just review from the plate tectonics lecture. We see rocks either preserving with normal polarity, meaning geographic north is pointing towards magnetic north, or reverse, which means geographic south is pointing towards magnetic north, and figure it out from there. So in the beginning of the Proterozoic, we had our single-celled organisms. These were mostly prokaryotes, so there were some eukaryotic single-celled organism. This is one. It is most certainly not a balloon animal. It is actually a fossil. We start to move towards multicellular life. This is a fossil found in Gabon, Africa in 2008. All right, so this part of Africa was only recently recognized or recently discovered to have these types of fossils, so we're starting to get more and more information from them rapidly at this point. Now, we're not sure what these fossils are. We think they're pretty close to bacteria, but we'll have a better answer for you in the next couple of years. So now we start moving into more complex types of algae. You don't need to remember the years that we find this. I'll never ask you when we find undifferentiated algae in the geologic record. Okay, but you can see here that we're moving towards the present day. So we're 1.7 billion years ago to 1.2 billion years ago. We're almost to modern day already. This is going to bring us to a pretty wild time on Earth's history. So when Earth gets that first couple hits of oxygen, it's going to go a little bananas, okay? There's positive and negative feedback loops on Earth, and when we have these positive and negative feedback loops, sometimes they can get a little out of control. So I'm going to tell you a hypothesis. I hypothesize that at one point in Earth's history, we had extensive glaciation on Earth, covering almost the entire Earth. How would we know? Okay, well, first, I would expect to see glacial rocks found everywhere on all the major land masses. Do you remember Alfred Wegener being confused by that? He found land masses or glacial rocks everywhere, like India, near the equator, in Africa, Australia, places where glaciers really didn't belong. Well, this is where they're from. All right. So here is a glacial rock. You can tell it's a glacial rock because you've got these giant hunks of pebbles thrown in piles of much smaller grain stuff. Those are glacial rocks. So we also have really high levels of C13, meaning low temperatures. Now, before I get ahead of myself, if Earth was mostly frozen over, would you expect an extinction? You probably would. However, we really don't see that. Some things do go extinct, but we don't really see a huge extinction like we think we would. So we originally called this idea snowball Earth, that the entire Earth was frozen over, and we thought it was a pretty good idea for a long time. However, we're starting to think that maybe we overestimated the evidence. So maybe it wasn't totally frozen over. Because things like stromatolites, these little blobs here, remember those microbial mats, they didn't die out either. So we'll call this snowball Earth for now, but it could really be slushball Earth. But whatever happened, we have the concept in play of runaway albedo. So albedo is the amount that a surface can reflect, or the amount of sunlight a certain surface can reflect. So what this means is ice has a really high albedo, okay? So ice reflects most of the sunlight instead of absorbing it. That's how ice stays cold. So if you ever go skiing or if you ever go hiking in the woods during a snowstorm, obviously this is not in Florida, you'll see that ice reflects that sunlight. It actually can hurt your eyes to look at it, okay? But on the other hand, we have things like earth, just like straight up dirt. If you've ever walked on dirt during the summertime in the hot sun, you'll know that the dirt can actually absorb sunlight and it doesn't really reflect it. It absorbs that sunlight. So if, let's take a second here to say this again. Ice has a high albedo, meaning sunlight reflects off of it. It is not absorbed, meaning ice does not grow warmer, meaning it would melt. Land has a low albedo, meaning it absorbs that water. It doesn't reflect it. the water is kind of in the middle. Water can absorb light, but it also does reflect it. That's why when you go swimming, you should definitely wear sunscreen because it can reflect a lot of that water as well, or that sunlight as well. So if sunlight isn't absorbed at the North and South Pole, all right, because there's ice there, that temperature of the ice will not get warmer. It will in fact get colder. When we have a colder temperature at the North and South Pole, we'll form more ice. If we form more ice, we're going to reflect more of that sunlight, which means it's going to get colder, which means we're going to form more ice. Do you see where I'm going with this? This is quite literally not, pun is not necessarily intended, a snowball effect here. All right. So as we get more ice, we're going to lower the temperature of Earth, which is going to force us to get more ice, which is going to force us to lower the temperature of Earth. So eventually, we'll see this system where ice continues to grow across the Earth. All right. So it will just keep continuing. And I know I've given you like 1,000 slides. My apologies here. So everyone agrees that there was probably a lot of glacial rocks during this time. So we probably call it slush ball Earth. So this is what it would have looked like. It just would have been a really cold time in Earth's history. And that's pretty common. We've seen it before, and we'll likely see it again in quite a few million years. But this had to do a lot with the breakup of Rodinia. So during the breakup, a lot of things were going on. So again, let's go back to our cookie analogy. Rodinia is a supercontinent, and if we take a giant cookie and break it up with a hammer, as a geologist might do, you'd see a lot of crumbs going off. And when we have all those crumbs in the ocean, they're going to be helping life flourish, nutrients, okay? So all that rock weathering is going to be going into continental breakup, and it's going to be burying a lot of that organic carbon. So what we're going to see at this point, here is our great oxygenation event, but where do you see the glaciations, the snowball or slush ball Earth? Anywhere we see more carbon-13, because we're burying all of that carbon. Temperatures are dropping, all right? So this is exactly where it's happening. Good work. So we're almost done, I promise. But right about this time, we have our first true animal fossil. Here's an embryo. It has kind of a six-fold symmetry, so it's not closely related to you. It's probably more closely related to an arthropod, like a bug or a spider or something like that. All right? And animals will show up once we get to the Phanerozoic, but we're not really going to talk about them today. But all of the dates here are changing rapidly. So a lot of the dates I give you may not be accurate in even just like a couple of years. So I'm going to show you some examples, some early fossils, but just know that the dates I give you might be wrong soon, okay? And just remember that oxygen levels are going to stay very low for a long time. But many animals, like sponges, don't really need that much oxygen to breathe. They just kind of sit there and do nothing. So here are some embryos from a very famous place in China called the Doshantou Formation. These are some of the most beautiful fossils. These are published by Zhao et al. In 1998. So Zhao is a paleontologist up in Virginia Tech. All right, but these are those embryos. Absolutely beautiful. We also find sponges, animals, from the Doshantou about the same time as the embryos. All right, so all these little black smudges are pieces of sponge bodies. And you might be thinking, okay, sponges are kind of boring. But sponges can do something you can't. If you put a sponge in a blender and turn it on, it can actually put itself back together later. You cannot do that. Please do not try. Okay? So why did complex life take so long to form? We've been talking about Earth history for quite literally billions of years now. So why is it taking so long for us to just get to sponges? Well, as you can imagine, Earth in the very beginning, in the Hadean, was super hot, meteorite impacts, everything was lava. It was really not easy to survive. There were lower levels of oxygen, and as soon as we get some oxygen, there's giant glaciation periods that make it really difficult for life to form. So, as you can imagine, drastically changing environments probably slowed down the evolution of advanced life. So here are your review questions. Please go through these on your own. I'm sure you're tired of listening to me talk for now, and I will see you next week. Thank you for listening.